So I talked about, oh no, that didn't convert well. So here's the left border, and then this is the right border over here. So these are just sequences that the bacteria recognizes to define the ends of that region. So they're not genes, they're just sequences. So here, DNA sequence that defines each end of the part of the DNA that gets transferred into the plant. And then we have these auxin and cytokinin. Those are both plant hormones. So together, these genes for plant hormone biosynthesis are what promote that tumor formation, which is that crown gall tumor you see that's characteristic of the agrobacterial infection. So those genes are being integrated into the plant genome, now forcing the plant to make these hormones at high concentration, forcing the plant to make that big tumor, which is a great place for the bacteria to live. And then this last thing down here, this is food for the bacteria. So what the bacteria has done is taken these genes that cause the formation of a tumor and a gene for making bacteria food. No cost to the bacteria, the plant is doing all of the work. So free room and board to the bacteria at no cost to the bacteria, but certainly plenty of cost to the plant because the plant is now putting energy into this uncontrolled cell growth, making these hormones, making this bacteria food. And these plants do actually stay alive, which is what the bacteria wants to keep, right? Plant needs to stay alive in order to keep producing food for the bacteria. So definitely a parasitic relationship that benefits the bacteria and does nothing good for the plant. So knowing now what gets transformed into the plant, are there other questions about these components before I go on? Okay, so what I'm going to ask you to do, so you've seen, hopefully, this is the plasmid. This is a circular piece of DNA, which is what Jeff was talking about last week with some cloning techniques, right? So you can amplify genes of interest, regions of interest. You can use restriction enzymes to cut plasmids. You can mix those two things together to make a plasmid that you want. So, and we know that these bacteria have invented a great gene transfer system for getting DNA into the plant genome. So what I want you to do is take a look at this, and in your groups, talk about what you would modify here, what you would take out, what you would keep, what you would add in order to get a trait that you're interested into a plant. Right, so we're taking the system the bacteria invented, and we're going to modify it to get a new trait into the plant that we couldn't do by breeding because the variation wasn't there and never happened by random mutagenesis. So some of the things I want you to think about are you need to be working with cells that get transformed. So if you think about the bacterial transformation you did last week in lab, thinking about the predictions you made about what was going to be transformed and what wasn't. So keep things like that in mind. Keep a trait in mind. Keep in mind things that you probably wouldn't want growing in your corn, out in your cornfield. So in your groups, change this part of that TI plasmid to now be able to introduce a trait that you care about into a plant.